Okay. Well, we'll lighten the mood with some great biology. So it's my pleasure, right? That's the biology keeps us going. It's my pleasure to introduce today's speaker, Maxine Zylberberg, who's coming to us from UCSF, where she's currently a postdoc in Joe DeRisi's lab. Before that, she was at the Cal Academy for 1 year as a postdoc. And before that, did her PhD at UC Davis, working with Tom Hahn. And then before that was at Amherst as an undergraduate, where she worked, I saw, with Paul Ewald. So she has a long-standing interest in microbes, pathogens, and their hosts, and in disease ecology. Most recently, she's been working on deep sequencing of metagenomic sequencing of viruses in birds, and that's what we're going to hear about today. So I'm looking forward to that. Maxine has also applied to be a postdoc here through the Miller postdoc and UC President's postdoc. So with any luck, we'll have you here in the MVZ next year. We'll keep our fingers crossed for that. Every week, Lelania asks speakers to provide some fun facts about them. And Maxine, I think, has won the competition here with the most interesting fun fact of any that I've read over the last 3 years. Her fun fact is, I've shared a phone booth with a lion. So whatever else you tell us about today, I hope you fill us in on that interesting observation. She also says, since the beginning of my scientific studies, I've been chased and or bitten by over 24 animals, including a sea crate, which luckily did not catch up with me, and a pelican, which did. So with that, I give you Maxine. Thank you very much.
Thank you. I guess I have to explain the lion.
Absolutely.
It was for lack of words that I said phone booth. I was actually in South America, where they have these tiny little shops called telefonicas where you can go to use a public phone. And I went into a little one where they had a couple counters set up with phones, and I was waiting in line. I was very busy. And a guy walked in behind me, and I turned to see, and he had a lion on a leash. And then I got a phone, and he got a phone, so we sat down next to each other at the counter with his lion. And afterwards, I asked to pet the lion, and I did. Because how often can you get to pet a lion, you know? Anyway, so today I'm going to be talking to you about the search for the cause of avian keratin disorder, which is an epidemic of beak deformities in black-capped chickadees. But first, why should you even care about disease in wildlife? And my first pitch to you is from a purely selfish perspective. About 70% of emerging diseases that we humans deal with come from animals, or have animals as a key part of their infectious cycle. And so here you can see a map of many of the recent emerging infectious diseases that have come out in the last few years. Every disease in red is one that is zoonotic, or has part of its cycle in an animal, or has come from animals. So we're talking about a lot of scary diseases, and a lot of diseases that we are trying to control as humans. So, for example, HIV originated in wildlife. Hantavirus, which is a really big deal here in California, routinely jumps from wild rodents into people and can often be deadly. And Zika virus, which we've been hearing a lot about in the news, of course, is transmitted by mosquitoes, and may be coming to the Bay Area soon. I think it's predicted to be in LA within a year. So from a purely selfish perspective, it's really important that we understand how and why diseases are spreading in wildlife. Now, from a conservation perspective, it's also important. Diseases have been increasingly linked with population declines and extinctions across taxa and throughout the world. So, for example, the last wild population of black-footed ferrets was driven extinct by canine distemper virus. Of course, amphibians globally are experiencing catastrophic declines due to chytrid fungus. And as a bird person, close to my heart are the Hawaiian birds, and many of these birds have experienced either declines or species extinctions as a result of introduced avian malaria and avian pox virus. And so both for public health and conservation reasons, it's really important for us to know what diseases are out there and why they're emerging. And this is what disease ecology seeks to do. So disease ecology seeks to understand how pathogens, and that's the bacteria, viruses, fungi that cause infectious disease, hosts, such as animals or humans, and the environment interact to determine pathogen dynamics. And so this could be, for example, understanding how host susceptibility to a pathogen affects dynamics, how the pathogen and environment interact to determine the severity of the pathogen and the impact the pathogen has on the host species. Questions like this, all with the goal of understanding the interactions of these factors and why pathogens spread as they do. So this is pretty abstract, so I will give you just a few quick examples from work that I've done to help put this in perspective. So, for example, I've studied how immune function, and this is just a little picture of an immune assay, how immune function varies in wild birds and how that influences susceptibility of the Galapagos avifauna to introduce avian pox virus, the same virus that was problematic in Hawaii. I've studied how variation in the endocrine system of hosts can actually impact and possibly even drive annual cycles in blood parasite emergence. That was worked in red crossbills. Often I get this question, this is a red crossbill and that crossed beak is natural. This bird does not have avian keratin disorder. And I've looked at how variation in affiliative and aggressive behavior in finches can impact the transmission of a pathogen through populations. Taking a more environmental perspective, I've asked how agricultural practices can increase susceptibility of host individuals to disease, how climate variation can impact pathogen transmission, in this case, for example, by influencing the vectors that transmit pathogens like mosquitoes or flies. And work with Paul Ewald, how the impacts of changes in human sanitation can actually drive an increase in virulence of cholera. So all of these studies focus on understanding the complex interactions between pathogens, hosts, and their environment in order to better understand pathogen dynamics in a system. So that's all well and good. One of the most basic questions, and simple questions really, that you can ask when studying a disease is what causes it. And that's what I'm going to talk about for the rest of my talk in the case of avian keratin disorder in this black-capped chickadee you can see here. And it seems like a very simple question. In the case of avian keratin disorder, the answer has actually eluded scientists for over 2 decades. So people have been trying to figure out what is causing this disease without success. So what is avian keratin disorder really? Well, the definition is very broad, largely because we don't know much about it. Avian keratin disorder is defined as an epidemic of beak deformities, and it was first documented in black-capped chickadees in Alaska in the early 1990s. Here in box A, you can see a black-capped chickadee with a normal beak. This is what a chickadee should look like. It's got a nice short beak. There's a nice tight interface. The two straight top and bottom beak meet at a point. All these other individuals have avian keratin disorder. And what you can see is that it really varies in severity. And there's a lot of different deformities that are classified as avian keratin disorder. You could have just a little overgrowth of the bottom beak, a more extensive overgrowth of the top. In some cases, both bottom and top overgrow and cross. They can separate. And in the most extreme cases, you can actually get birds with a beak that is many times the normal length. So beaks, of course, are vital for birds. Not only do they use their beaks to eat, they use their beaks to handle their food, process their food. They use them to preen. So they use beaks to remove ectoparasites and stay healthy that way, and also to maintain the integrity of their feathers, which are vital for thermoregulation. So these black-capped chickadees, pictured here, are all in Alaska, and they stay in Alaska year-round. So you can imagine that staying warm is particularly important for these birds to survive. And so as a result of these massive beak deformities, these birds have diminished foraging efficiency, they are unable to preen, they have very heavy ectoparasite loads, their feathers are just a mess. So, and they have poor body condition and are also more susceptible to predators. So these guys suffer really high mortality rates, as you can imagine. So, more bad news today. Now that doesn't mean they all die. A lot of birds have been seen to survive with avian keratin disorder, and this is especially true because they're a backyard feeder bird. And there are lots of backyard feeders around. The birds with avian keratin disorder are especially frequent visitors at those feeders. So the supplemental food allows them to survive, but of course it also raises concerns about disease spread to other individuals. And I should also say some individuals recover, which is great, it's hopeful. Some of these birds with really long beaks are actually recaptured. A handful of these birds with really long beaks, I should say, have been recaptured in subsequent years with normal beaks. And so, for those of you who are bird people, you probably already know this, but the bird beak grows constantly throughout the bird's life, much like our nails and fingernails. And so, our hair and fingernails. And it's through constant roughage and usage of the beak that it stays the proper length. And so this suggests that if for whatever reason these beaks are growing, if they stop growing, the bird is able to recover and return to having a normal beak. Here is what the section of the beak would look like. There's bone at the base. It's covered by connective tissue and blood vessels, a germinative layer of growing keratin cells, which then move outward and become cornified. And that cornified layer forms this outer sheath of the beak. What you can see here are columns showing a picture, an X-ray, and histopathology, all of the same bird. This first bird is a normal individual with no avian keratin disorder. And going this way, they have increasingly severe avian keratin disorder. And so what you can see with the X-ray, the arrows are pointing to the tip of the beak, to the tip of the bone, is that the bone actually looks pretty constant throughout these individuals. There seems to be very little deformity of the bone. I think the light is not good for this slide here, but here you can see this white layer that is the cornified keratin. In a normal beak, it stops here, and then what you probably can't see, maybe in this last one you can see it, it's that cornified layer that really overgrows and causes the deformity, with pink intrusions which represent actively grown keratin cells kind of going into that beak. So it's those keratin cells that are first of all becoming disorganized, and actively growing cells are intruding into the cornified layer, and it's also that area that's overgrowing. So thus the name avian keratin disorder. As I mentioned, it first appeared in the early 90s. The first two cases noted of avian keratin disorder were in western and south central Alaska in 1991 and 1992. Since then, it's been observed in thousands of black-capped chickadees in Alaska. What's more, it appears to be spreading south. When it was tracked over time, it appeared to be spreading south and east. So this is a disorder that is actively spreading spatially in black-capped chickadees. Most of the work has been done in Alaska, so the number of sightings here should not mislead you into thinking there's necessarily less keratin disorder there, but these are opportunistic data points. And what you can see is really a lot of the chickadee range seems to have avian keratin disorder as of 2009. 6.5% of the black-capped chickadee population has avian keratin disorder, exhibits avian keratin disorder at any one time. It fluctuates annually, sometimes it's as low as 4% and sometimes as high as 10%. But really, that's the big number for a disease that is largely deadly to these birds. So a lot of birds are being affected. and it's spreading. And it's not just black-capped chickadees. It actually, these similar deformities have been seen during the same period in a wide range of birds in the United States. It's especially prevalent in northwestern crows. 17% of northwestern crows have these beak deformities. It's also appears to be very common in raptors. There's not good data published on this, but the raptor community actually started observing the very similar deformities at just a few years after avian keratin disorder was documented, and they started calling it long-billed syndrome. So they were seeing a large number of a variety of raptors, but especially red-tailed hawks, with beak deformities very similar to avian keratin disorder, with many of the same characteristics. And red-breasted nuthatches also seem to have a number of individuals affected. So it's not limited to one type of bird. These are seabirds, they're raptors, they're songbirds. There really isn't a unifying characteristic among these species. And it's not just in the US. About 2 years ago, they started to document large numbers of similar deformities in the United Kingdom. So it really does appear to be spreading spatially. There have actually been these types of deformities documented in 36 species of bird in the UK, but I think I kind of go with the number 12 at this point because there are 12 species where really high numbers of these deformities have been documented. So, with just a few documented cases, it's hard to know if it's a naturally occurring deformity or part of this kind of epidemic of deformities. So what is causing this? It's affecting a lot of species, it's spreading. What could be doing it? So there are a number of known causes of beak deformities in birds from the veterinary literature, from the ecological literature. Environmental toxins such as selenium and PCBs cause beak deformities. They tend to look different. They tend to be present in nestlings or hatchlings, whereas avian keratin disorder is almost exclusively present in adults. Nutrient deficiencies can cause beak deformities. Again, they often look different from what we're seeing in avian keratin disorder. And there are several pathogens that have been associated not necessarily with beak deformities of this style, but with beak lesions or beak pathologies of one type or another, including 3 viruses: polyomavirus, poxvirus, and circovirus. So these were the go-to candidates. And my avian keratin disorder was first documented by my collaborators, Caroline Van Hemert and Colleen Handel in Alaska at the USGS. They investigated each one of these causes in turn. Despite the different pathologies, they went after each one, and they ruled each one out. They did not find evidence of any of these causes in their birds with avian keratin disorder. So that's where I came in. Because of the spatial spread, the manner of spread of the disease, and also because of the variety of species affected, this was really highly suggestive of this disease being infectious. Now, bacteria and fungi you can usually see microscopically. A number of assays were done trying to grow bacteria or fungi, and there was no evidence whatsoever that either bacteria or fungi were associated with this pathology. So we really suspected a virus as being the causative agent of this disease. However, we had no idea what virus it was. We had an idea of 3 viruses that it probably was not, that had already been ruled out. And so what I did is I used the Illumina Next Generation sequencing platform to try and find any pathogen associated with this disease. For those of you who are not familiar with metagenomic sequencing, the power of metagenomic sequencing is it allows you to sequence everything in a sample all at once without having to have prior knowledge of what you're looking for. So I could take a sample of a bird beak from an individual with avian keratin disorder and sequence it and get information on the host genome, on the bacteria that was naturally occurring in its mouth, on maybe the last insect it ate before it was collected, and on any any virus that was in there. And the way this works is you basically take the sample and you chop up the DNA or RNA, whichever you're interested in, or both, into little pieces. Oops. And you essentially stick those pieces onto a glass slide. Once they're on that glass slide, can you see anything there? No. So imagine you see a kind of a necklace, a pearl strand here that represents the strand of DNA or RNA that's been stuck there. And here is a complementary DNA strand that's being made to the original genetic material from the sample. And what you do is every time you add a nucleotide, you cause it to glow. And the color it glows depends on the nucleotide, which of the 4 nucleotides is added. And in that way, you're able to image this with a computer and find out what the sequence of your DNA is. And in fact, you do it all at the same time, and you get information on millions of sequences all at once. So I did this using RNA that I extracted from 10 individuals with avian keratin disorder. I extracted the RNA from Beaks, I pooled them, and I put it through this system. What I ended up with was 50 million sequences, or short reads of DNA. And they were really short. They were only 50 nucleotides long. And basically, what that looks like is, I pictured it here. Again, the color's not showing up. Imagine that in addition to these gray and red lines, there are lots and lots of blue lines. And those blue lines represent the avian host. And there's tons of that. Really, that's most of what's there, and it's not what we want. Mixed in with that are some reads, represented by these gray lines that might be bacteria or other organisms. And then a handful are going to be virus, potentially. That's what I was hoping, represented by those red lines. And so using bioinformatic tools, I am able to stitch together each of those reads that matches up. Here, each row is representing one read, and you can see that with somewhat overlapping but not completely overlapping reads, you're able to build out a bigger and bigger genetic sequence. And then compare those sequences to NCBI and other databases with all known genomes. For those of you who do metagenomic sequencing, here was my basic pipeline. It's very simple. We're doing much more sophisticated, complicated, more efficient searches now. But for this data, I just removed low quality sequence. I used BLASTN to align reads to chicken and zebrafinch genomes, is what I used at the time, to pull out as much bird as I could. It would have been better to use a chickadee genome, but I didn't have it. So I removed as much host genome as I could, and then I used the Trinity de novo contig assembly to assemble up the remaining reads into contigs. Then I used, I compared those sequences to nucleotides in GenBank, which is really targeted for identifying known pathogens. And then also I converted to amino acid sequence and compared that to existing amino acid sequences, which is better at identifying novel pathogens that perhaps have more nucleotide variation but have some conserved amino acid sequences. And ultimately, after all this was done, I had assembled a bunch of bird, a bunch of random plant material, whatnot, and the full genome of one virus, and it was a coronavirus. Now, and here it is. So by comparing the genome that I had to existing coronavirus genomes, I was able to figure out essentially what the genome organization of this virus was. Now, coronaviruses are RNA viruses, and their entire genome codes for a single polyprotein. So it's a single protein that the virus then chops up into actual functional protein units. And at the 5' end of that protein sequence are proteins that form the outer capsid of the virus, and these are often fairly well conserved across coronaviruses. And then all the rest of these are involved in viral replication. And actually, so one thing that was actually quite challenging in assembling this this genome is that I got the full genome right away, but I thought I was missing a big chunk of the genome because this protein, in all the closest relatives of this coronavirus, is 1,500 nucleotides longer. I think mine was something like 60 nucleotides, and usually it's 1,600 nucleotides. And so I spent a long time looking for the missing sequence until I finally figured out that no, this was it. So this coronavirus is missing a big chunk compared to its known relatives. These codes here tell you the amino acids it was cleaved at. It doesn't really matter what they are, but they are different from the typical, highly conserved coronavirus cleavage pattern. So this graph shows you the coverage all along the entire protein, the number of reads in my data that support each one of the nucleotide calls across this whole genome. So the support is pretty good. I had about 140x coverage on average across the genome. There are no big missing holes. But what was striking was that this virus is only about 28% similar to its closest known relative. So it's missing a big chunk, the cleavage sites are different, and it's really different from pretty much any other coronavirus we have seen. So that was really interesting. I should also point out that bioinformatically assembling a genome, as you can imagine, can be really error-prone. So I didn't just take the computer's word for it, I actually went across and Sanger sequenced and confirmed the full polyprotein coding region of the virus. So we know that this virus is what we think it is. So here is a tree, a phylogenetic tree, showing all the fully sequenced coronaviruses that are known to people, color-coded by host. So for those of you who are not as familiar with coronaviruses, some examples of coronaviruses that you probably know are the common cold, polio, foot and mouth disease, which of course is big in England. They do a lot of culling thanks to that, in cattle. And those are all, well, those will all be in blue, so I'm a million. In orange are about 24 coronaviruses that have been isolated from avian species. Except for 2, all of these viruses have been isolated from poultry and are typically associated with stunted growth in juveniles, with less laying in hens, so not very exciting. There are 2 viruses isolated from wild birds, and nothing is known about them. They were just opportunistically found in sampled birds. And there is one virus, which is from pigs, called Seneca Valley virus, which causes keratin overgrowth. So that's intriguing, but really, ultimately, it's not a very informative group. It doesn't confirm or tell us much about whether we think this virus is causing avian keratin disorder. What it does do, and which is important, is that Poesi virus, which is what we're calling the coronavirus we found in black-capped chickadees, clusters here with the megriviruses, which is a nice big group of coronaviruses isolated from birds. So what this analysis does do is it gives us some confidence that the virus that I found is a bird virus, and it's not something that was infecting an insect or just happened to be in these birds. But really, we want to know, is this virus causing avian keratin disorder? So you may remember I told you when I did the deep sequencing, I actually pooled samples from 10 individuals. So I didn't even know at this point if this virus was present in one bird or multiple birds from my sample. So I designed specific primers, PCR primers, to target Poesi virus and start to test individuals, both with avian keratin disorder and with normal beaks for Poesi virus. And to start with, I only had 28 beak samples. I focused on beak samples to begin with because the beak is actually the only pathology in these birds was seen in the beak. Otherwise, for necropsies, they appear to be totally normal. So I focused on the beak for the sequencing. But that also meant that the birds had to be sacrificed, and we had a fairly limited sample as a result. However, we can still look at the correlation between avian keratin disorder and Poesi virus. So what you're seeing here is for 28 individuals that were sampled, their beak length on the x-axis, and avian keratin disorder, for the purposes of our study, was defined as a beak length of 8.1 millimeters or longer. So you'll notice this goes to 40.3. This is not to scale, but I scrunched it to get the data in there. Or an individual whose beak was shorter, but who had evidence of hyperkeratosis at the cellular level. And so that's this one individual here. They actually had a beak of 8.1 millimeters, so it wasn't quite at the cutoff, but they did exhibit hyperkeratosis. So these individuals all had AKD, and they were all infected with poecivirus. So infected individuals are shown in orange. I also had 9 individuals who had apparently normal beaks, and of those 9 individuals, 2 individuals had poecivirus. So this suggested a pretty good correlation between the virus and the pathology, but it's a very small sample size, so it needs to be taken with a grain of salt. At the same time, I was looking at viral tropism, and so I wanted to see, okay, well, the pathology is all in the beak, but can we see this virus elsewhere in the birds? And so the first 2 birds I tested, something really interesting popped out, and that was that viral load was actually higher in the cloaca than in the beak. Oh, and I should tell you this viral load is the number of viral particles detected normalized to the number of host RNA, ribosomal RNA particles, so it is a normalized number. And so that was really, really exciting for us. It was not that surprising, because a lot of picornaviruses are transmitted via the fecal-oral route. But it was exciting. And so moving forward, when I tested the next 2 birds, we first took buccal, beak, and cloacal swabs to see whether we could actually detect the virus in a swab rather than in tissue sample. And you can see the data for those swabs here in orange is a buccal swab, and yellow is a cloacal swab. And so what you can see is actually the viral load was as high in the cloacal swab as in any tissue that we tested, which was great. What that meant, going forward, is that we could test cloacal swabs, and we could really increase our sample size. And so this is actually pretty new data. My collaborators collected, they collected 100 swabs. Some of those were not from chickadees and are not included here, or not from black cap chickadees, so they're not included here, just in the past few months. And so we were able to really increase our sample size to 115 individuals from 28. So here's the same graph. Here you can see the beak length. Oh, and I didn't point these guys out before, but just to remind you, here's a normal beak. Here is the farthest out beak here. This guy is a beak that was measured at 40.3 millimeters. And so you can see that's massive. That's coming down almost to its rib cage. Clearly, he's not going to be able to eat much or survive for very long. So here we have our individuals with avian keratin disorder. And with a substantially increased sample size, every individual with avian keratin disorder is still testing positive for poecivirus. They all have it. We have a much bigger sample size of control individuals, which is great, because we were really, really missing that. And so ultimately, of 88 control individuals, oh, sorry, wrong number, 3, actually about 3% were infected with poecivirus. So a very small percentage compared to 100%, or 27 out of 27 individuals with avian keratin disorder. So this is starting to be quite a good correlation between poecivirus and the presence of avian keratin disorder. So what's up with these 3 individuals? So they kind of could be problematic to the idea that poecivirus is causing this disease. One possibility is that this data is not based on full genomes of these viruses. This is based on targeted PCR primers that I designed to sequence the 200 nucleotide segment out of these viruses instead of the full 8,000 nucleotide virus. So it's just a tiny piece. So one possibility is that maybe these viruses are different from, maybe it's a different species of virus from what we're seeing causing poliovirus. So I actually, I sequenced the full genome of 2 of these 3 viruses to see, is this a different virus? Are we dealing with 2 different, perhaps, strains? So what you can see here is a tree of poliovirus, the full genome, full 8,000 nucleotide genome of poliovirus from 5 individuals with avian keratin disorder and 2 individuals who were normal, who did not have any signs of avian keratin disorder. And so, first of all, these across all these viruses, they're about 96.6% identical. So this is the same virus. It's not a different virus in these birds. And beyond that, the virus in the normal birds is clustering with the viruses from the affected birds. So it's the same virus. So that leaves us with the possibility that, 1, poliovirus may be infecting all these birds with avian keratin disorder, but it may not be causing it. It could be that it's an opportunistic infection that they're just much more prone to getting. Another possibility is that poliovirus is causing avian keratin disorder, but that these 3 individuals either are asymptomatic, which happens in many, many disease systems. They could be asymptomatic carriers. Or it could be that they just haven't developed the disease yet. So if you remember, there was that one individual with the 8.0 millimeter beak who was actually classified by our collaborators as being a normal control individual. And it wasn't until we went and did the histopathology to confirm that it was in fact normal at the cellular level that we saw no, it wasn't, that it was actually exhibiting a keratin overgrowth and it simply hadn't manifested yet as a beak disorder. So one of the striking things that I mentioned early on about avian keratin disorder is that it affects dozens of different species. And so one way to get at the question of whether we are confident that poliovirus may be causing avian keratin disorder is to see whether we can find the same virus in birds with deformed beaks of other species. Now, this is very, very recent work, and my sample sizes are teeny, teeny, tiny, so this is not conclusive at all, but it is interesting. So, So I recently received 2 individuals from Alaska, 2 red-breasted nuthatches that had severely overgrown beaks. This is one of them, and one had almost a severely overgrown beak. Quite impressive. 2 northwestern crows that had overgrown beaks, and 1 hairy woodpecker with an elongated beak. And actually, the hairy woodpecker that we got was not this one. If it had been pictured here, its beak would be elongated to about here. And it was in really, really horrible condition, and I thought about putting a picture in, but it was sad. So I tested each of those individuals for poliovirus, and every one of them was positive. So, you know, these were all birds from Alaska, so again, it could be just another a virus floating around in Alaska, but each one of these birds was positive for poliovirus. I also recently received, but have not yet tested, some samples from red-tailed hawks with long-bill syndrome, and several samples of boreal chickadees with elongated beaks. And so, that, like I said, this is something that I'm just starting to work on, and we're working to try and really increase our sample size of individuals with elongated beaks and also control individuals from these species, because of course it's really important to see whether the virus is present in normal individuals. That said, I did look at the relatedness of the viruses from these individuals, given the limited data that we did have. And it was interesting. You know, the viruses from different species seem to be clustering together. And that is despite when the sample was collected. So, for example, especially with the red-breasted nuthatches, the first one was collected in 1999 and stored frozen. The second one was collected in 2012. And so the viruses from these two individuals, despite being collected 13 years apart, were very similar and clustered together versus the black-capped chickadees that were actually all collected within that time period. But they appear to be separate and different. The red-breasted nuthatch virus appears to be about 92% similar to the chickadee virus. This is based on partial genomes, so I'm working on getting the full genome for each of these individuals to see how similar they really are. And the northwestern crows are about 91 and 93% similar to the chickadee viruses. So it's unclear yet kind of what that means for the viral ecology. And then here's the hairy woodpecker in there, mixed in there with the chickadees. So it's interesting both because it shows that there may be relatively few crossover events of viruses between the species, and also that there is a potential for some amount of pathogen-host coevolution. All right, so moving forward, what's the next step? Well, I hope I've convinced you that there's a pretty strong association between poEC virus and avian keratin disorder. They seem to be very closely associated. But, as with any disease, what you really need to do to show that a pathogen is causing the disease is you have to cause the disease. And so to this end, I spent more time than I want to think about trying to grow this picornavirus in many, many different ways in, I think about 7 different avian cell lines, mammalian cell lines. I actually went to Davis and collaborated with an expert there on growing picornaviruses to try and grow it in fertilized chicken eggs, and it wouldn't grow. Only about half of picornaviruses are culturable given our current method. So this, it wasn't terribly surprising, but it was frustrating. But so moving forward, we actually are working with collaborators and vets to try and design an infection experiment to co-house chickadees with avian keratin disorder in a very controlled setting with normal chickadees to see if we can really get poEC virus transmission between birds, to see how it's transmitted, and to see if birds that become infected with it develop elongated beaks. And that's really going to be key because knowing whether this virus causes the disease is going to allow us moving forward to start to do ecological studies to look at how is the virus transmitted, how easily is it transmitted, how quickly does it spread, and how does it spread between species. And so and what is the impact on host fitness and populations? And so those are going to be really key questions for understanding the effect that this virus might have on bird populations moving forward. And then I mentioned I'm starting to look at, I'm really excited about looking at differences in viruses between species, looking at differences in how species are impacted by this virus. Perhaps some birds are much more prone to developing disease as a result of infection, and to see whether there is some coevolution happening between different viruses and their hosts. So moving forward, I want to acknowledge my collaborators, Carolyn Van Hemert and Colleen Handel, who did a lot of the original work describing avian keratin disorder and who provided all the chickadee and other samples for this work. Jack Dumbacher from California Academy of Sciences, who was instrumental in getting this work up and running and collaborated with us on it. And Tariq Tuhan, who did a lot of the histopathology, which I didn't show you, but was really vital to confirming or denying which individuals were affected by avian keratin disorder or were normal. And of course, I'd want to acknowledge Joe, somewhere in this picture, and the whole Derizi lab, who are here on the BioHub deck, so we've got a fancy new deck, and they've all been very supportive and helpful throughout this project and funded sources. And with that, I'd love to take any questions.
This disease maybe somehow involved in evolving these crazy bill shapes of, I don't know, like some of them look like honey creepers and some of them look like crossbills. Is there any, I don't know, just, what do you know about the developmental biology of bills and how it might be related to same mechanisms of the disease.
I am not an expert in the developmental biology of bills in any way, but I would not expect that such a rapidly emerging and kind of deadly sort of disease, and one that really decreases host fitness so much by not allowing them to eat, not allowing them to feed their nestlings and whatnot, would be the driver behind crossbill beaks and honeycreepers.
Although,
But I don't know.
I mean, it just seems like if
yeah.
it were somehow able to be incorporated in the genome, and it did increase their feeding frequency because they were able to do something
Yeah.
new.
Maybe kind of a milder,
I don't know.
milder form of the virus. Yeah. Okay. This virus in particular, actually, the overgrown part of the beak is incredibly brittle, and so it snaps off very
Oh.
easily. Whereas the crossbill beak is very hard. Of course, you have to pry open those cones. But yeah, entirely possible. Yes.
So, it's two sort of related questions. Are birds with longer, more deformed beaks older than birds with less deformed beaks? And is there a correlation between viral load and beak length?
We don't have good data on the age of a lot of these individuals, so I don't know if the longer beaks are necessarily older, but I would guess not. And the reason I'm saying that is because some of these birds are captured with normal beaks in June, and when they're recaptured 6 months later, they were massively overloaded. So these beaks are elongating very quickly. It's not kind of slow over time. And no, viral load does not appear to correlate with the severity of the beak deformity. That said, the viral load that I'm sampling in individuals is from a single time point when the individual is collected. And so, it may be entirely possible that if you were able to have the individual in captivity throughout its sickness and sample throughout, that maybe the peak viral load is higher in birds with longer beaks. I don't know, but that would be something that would be really interesting.
Sort of when you get the disease affects how the phenotype emerges.
Right.
Yeah, I'm just curious to know if you looked at what genes are present in the virus genome and if any of those might be candidates for causing the deformities on the cellular level.
So that the genome I showed you of the virus showed every protein. I think there were, gosh, I should know this, I think there were 9. Okay. And the capsid proteins are very well understood, and the 3' proteins, the polymerases are very well understood, and those 3 middle proteins, we don't really know what they do. And so one of those, of course, of those middle proteins that we don't really know what it's doing, is one of the ones that was really, really different and much shorter than the related viruses. And then I think also if I go back, you'll see that some of those, there. So these areas, especially this 2A and 2B protein, are some of the least similar areas to related viruses. These areas are pretty darn conserved because that's what actually replicates the virus. But yeah, maybe something's going on there. Maybe that's the area that's doing it, but I don't know.
You said that the disease only seems to be affecting adults. You actually sampled juveniles and
Yes.
looked for the presence.
Yes.
Okay.
So the study in Alaska where a lot of people come, they have nest boxes, and so they actually monitor breeding and look at the hatchlings and nestlings. And it's been observed, I believe, the earliest individual it was observed in, I think, was 6 months old, but it's only been observed in a handful of individuals that are that young, and otherwise it's adults.
So I was thinking about what are the potential routes of transmission, and so I've never heard of a bird sexually transmitted disease, but given the coigal results, got me thinking about that, and also wondering if it's not affecting the juveniles. It would be interesting to see if you tested fecal samples for the virus, that might rule out like any like environmental transmission. Because if you're
Absolutely.
like nesting and juveniles aren't getting it, then maybe it's specifically just among breeding adults.
Yeah, I think that's a good idea, and we're working on increasing our sample sizes now with the swabs kind of across all groups.
Did you do fecal samples before? Or?
I have not done fecal
Okay.
samples, only swabs.
The phylogeny of the virus makes it look like there's not a lot of horizontal transmission between species, maybe with the exception of the woodpecker. But and so that suggests that it's been in these birds for a while, and there are a lot of people bird watching all the time. What's caused the outbreak?
I don't know. That's kind of what we're all wondering, and to figure it out, we need to know what's causing it. As far as the outbreak goes, one thing that they have looked at is to see whether it's a sampling thing. Are people looking for it? And so in Alaska, they did do some work looking at museum specimens. They also went back. It was first noticed as part of a long-term study of chickadees, and so they actually went back through all their records and looked to see if there had been high beak deformities that they were missing, and there weren't. It really was this rapid increase in beak deformities. So I don't know what's causing it. Is it, you think, well, is it some sort of environmental contaminant that's making them more susceptible? But something like that you would think would be perhaps contained by a watershed, perhaps unique to a specific type of species or specific food, bird that eats a specific type of food, and that none of those are true, which makes it seem like maybe it's a newly evolved virus. Perhaps it's something that came out quickly and then stayed within bird lines. I can only speculate.
And it's striking that it's appeared recently in unrelated species of birds.
Yes, absolutely. And it does really appear to be spreading. So in northwestern crows, they show the same pattern of deformities, first being seen in Alaska and the Pacific Northwest, and then spreading south and east. And interestingly, in crows, it showed that same pattern, but it happened much faster. And crows, of course, have a much larger range. The chickadees basically stay in one spot their whole lives, and the crows are moving much larger distances. So that does make it seem like it's something that's spreading, and it's as a result of contact that it's moving. But why it happened now, I don't know.
I'm wondering if you thought of looking at the total flora of the birds that are infected, because if the coronavirus is a secondary consequence of some other agent, then maybe you would expect the whole flora to display some sort of imbalance or dysbiosis or something.
I have not looked at that. I think that we've talked about it. I would expect to see differences. I mean, their diet's changing, their behavior's changing.
That's
they have a higher
true, too,
parasite
yeah.
load. There have to be changes. Would you like to do that?
Yeah.
Great. Okay.
Maybe you'll take one more question.
I think there's one.
Sure.
Oh.
Yeah, there are two more. Go ahead. Okay.
Oh, okay.
So some of the effects of viruses, of course, are known to have to do with their spread, like thinking of sneezing and diarrhea in humans. But presumably this is just a fluke. This overgrowth of keratin is just a fluke. So is that kind of the thinking? I mean, it's a very odd pathogenic, pathological impact of the virus.
Right.
I can't think of anything else similar to
Absolutely,
that.
absolutely. It's very odd. The documented pathologies, for example, toxins and nutrient deficiencies, often are things that affect the liver, and then because of the physiology of beak growth, the impacts on the liver are manifested in beak growth. So perhaps something like that is going on here. Levels weren't particularly high in the liver, however, so I don't know. But yeah, it doesn't seem like the beak growth necessarily would help spread it. The one thing is that, for example, the birds that have overgrown beaks are less efficient at foraging, and so they come much more opportunistically to, for example, feeders. Northwestern, the crows often feed at garbage dumps. And so both chickadees and crows are using human sources of food, and so they're being artificially concentrated, and especially impacted birds. So, maybe that's something about the virus pathology that would affect spread, but really only in the presence of humans, so I don't know. I don't know what to think about that.
All right. Well, let's thank Maxine again.